Chapter number 3, Plant Kingdom. In previous chapter we looked at the broad classification of living organisms under the system proposed by Whittaker 1969, wherein he suggests the five kingdom classification: Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with further classification within kingdom Plantae, properly known as the plant kingdom. We must stress hence that our understanding of the plant kingdom has changed over time. Fungi and member of Monera and Protista having cell wall have now been excluded from Plantae. The earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm and angiosperm under Plantae. Let us look at classification within angiosperm to understand some of the concerns that influenced the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number, and shape of leaves, etc. They are based mainly on vegetative characters or on the endogenous structure system given by Linnaeus. Such systems were artificial. They are separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification system developed, which were based on natural affinities among the organisms and consider not only the external features but also internal features like structure, anatomy, embryology, and phytochemistry. Such a classification of flowering plant was given by George Bentham and Joseph Dalton Hooker. A phylogenetic classification system based on evolutionary relationship between the various organisms are capable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that used to chemical constituents of plants to resolve confusion, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll bearing simple thyroid autotrophic and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and wood. Some of them also occurs in association with fungi, lichen and animals, example are slothweed. The form and size of algae is highly variable, ranging from colonial form like volvox and the filamentous form like Ulothrix and Spirogera. A few of marine forms such as kelp form massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different type of spores. The most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Ulothrix or non-flagellated non-motile but similar in size as in Asparagus. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in a species of Eudorina is termed as an isogamous. Fusion between one large non-motile static female gametes and a smaller and motile male gamete is termed oogamous. Volvox and fucus is an example. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy rich compounds which form the basis of the food cycle of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, water holding substance, that is algin, brown algae, and kerosene, red algae, which are used commercially. Agar, one of the commercial products obtained from Xylidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in protein, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, Rhodophyceae. Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, filamentous. They are usually green grass due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral, ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplasts. Pyrenoids contain protein beside starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes by fragmentation or by formation of different type of spores. Asexual reproduction is by a flagellated zoospore produced in zoosporangia. The sexual reproduction shows considerable variation in the type of and formation of sex cell, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Urothrix, Spirogyra, and Cara. Phaeophyceae. The member of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms ectocarpus to profusely branched form as represented by kelp, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown, depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminarian, or mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk and the stipe and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction is most brown algae is by a biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, and oogamous. Union of gametes may take place in water or within the Oogonium oogamous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common form are ectocarpus, dictyota, laminaria, sargassum, and fucus. Rhodophyceae. The member of Rhodophyceae are commonly called red algae because of the predominance of red pigment are phycoerythrin in their body. Majority of the red algae are marine with greater concentration found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depth in ocean where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually via non-motile spores and sexually via non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common members are Polysiphonia, Porphyra, Gracilaria, and Zelidium. Table 3.1 Divisions of algae and their main characteristics. Class Chlorophyceae. Common name green algae. Major pigment chlorophyll A, P. Stored food starch, cell wall, cellulose. Flagella 2-8 equal apical. Habitat freshwater, brackish water, and salt water. Class Phaeophyceae. Common name brown algae. Pigment chlorophyll AC, fucoxanthin. Stored food mannitol, laminaria. Cell wall, cellulose, and algin. Flagella 2 unequal lateral. Habitat freshwater, rare brackish water, salt water. Class Rhodophyceae. Common name red algae. Pigment chlorophyll A, D, and phycoerythrin. Food storage Floridian starch, cell wall, cellulose, pectin, and polysulfate esters. Flagella absent. Habitat freshwater in some brackish water, salt water mostly. 3.2 Bryophytes. Bryophytes include the various mosses and liverwort that are found commonly growing in most moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrated or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structure. The main plant body of the bryophytes is haploid. It produces gametes, hence is called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoids. The female sex organ called archegonia is flask-shaped and produced a single egg. The antherozoids are released into water where they come in contact with archegonia. An antherozoid fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous animals, birds, and other animal species of sphagnum. A moss provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichen, are the first organisms to colonize, colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plant. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as bank of streams, mossy ground, damp soil, bark of trees, and deep in woods. The plant body of liverworts is thalloid. Example, Mercantia. The thallus is dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structures. Asexual reproduction is in liverworts takes place by a fragmentation of thalli or by a formation of a specialized structure called zemi. Zemi are green multicellular asexual bud which develop in small receptacles called zema cup located on thalli. The zema becomes detached from the parent body and germinate to form new individual. During sexual reproduction, male and female sex organ are produced either on the same or on different thalli. The saprophyte is differentiated into a foot, seta, and capsule after meiosis. Spores are produced within the capsule. These spores germinate to form free-living gametophytes. Mosses. The predominant stage of the life cycle of moss is the gametophyte, which consists of two stages. The first stage is protonema stage, which develops directly from the spores. It is a creeping, green-branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema. As a lateral bud, they consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bear the sex organs. Vegetative reproduction in mosses by fragmentation and budding in secondary protonema. In sex reproduction, the sex organ antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consti- consisting of a food, septa, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule become the capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum, etc.